Hi everyone, my name is Habib. I'm a PhD student at Simon Fraser University. I'm going to present our project, Continuous Chromatin State Feature Annotation of the Human Epigenome. Semi-automated genome annotation methods take as input real-valued functional genomic data tracks like ChIP-seq and DNA-seq data that are defined over the genome, and they generate an annotation that summarizes the regulatory activity at each position. Examples of these methods are epigenome-ssm and segue. These methods segment the genome so that for each position, we have a single integer label assigned to it that represents the type of activity or functionality of that position. We can later use our biology knowledge to interpret these labels. For example, we say label 1 represents enhancers. This type of annotations have some limitations. We cannot represent the string of genomic elements. For example, if we have a promoter and a weak promoter, a discrete annotation need two different labels to assign to each of these elements. Also, we cannot represent elements that have multiple types of activities, the combinatorial elements. One example is intronic enhancers. These elements usually have marks of both transcription and regulation. And again, we need two different discrete labels to represent these elements. To address these issues, here we propose a continuous annotation strategy. So instead of assigning a single integer label to each position, we generate a vector of continuous features. So instead of saying that this position is an enhancer, we generate a vector of continuous features, and these vectors represent the degree to which this position acts as an enhancer. This type of annotation have some major advantages over discrete annotations. First of all, we preserve the underlying continuous nature of the input signal tracks. We can also capture the strength of the elements and handle positions with multiple types of activities. Also, continuous features are very useful in visualizations and other downstream analysis. This is the overview of our method. Like other Saga methods, we take as input signal tracks over the genome from different assays, and we generate a vector of continuous features for each position. To do this task, we use a non-negative state space model, which we call it epigenome-ssm. SSM is very similar to hidden Markov models. The difference is that hidden Markov models generate latent variables which are discrete, but latent variables in SSM are continuous. SSM has two major assumptions. The first one is that for position G, there is a latent vector alpha that encodes the chromatin state feature at that position. And the input signal tracks at position G, which we represent by YG, is actually generated by a linear combination of the latent vector at that position. Another assumption is that latent vectors at each position are generated as a result of a linear combination of latent vectors at the previous position. We did a bunch of evaluations to see how useful these continuous features are. And we have our method epigenome-ssm and a variant of our method, which we call epigenome-ssm non-neg. Epigenome-ssm non-neg has an extra constraint on the generated features to be non-negative. And we compare our method to well-known annotation methods like epigenome-ssm and segue. So here we use the annotations generated by each method and pass it to a linear regression model to predict gene expression and enhancer activity. And we found that annotations generated by epigenome-ssm are better predictive of gene expression and enhancer activity compared to annotations generated by epigenome and segue. We also found that these continuous features recapitulate known genome biology. So from this heat map, where we use segue annotations as our reference, uh, we can see that feature 1 is representing promoter activity and feature 5 is representing enhancer activities. We can see the same in these scatter plots where feature 1 is having high values at promoter positions and feature 5 is having high values at enhancer positions. And down there, we can see that feature 1 is having a strong correlation with the expression values of the genes. To summarize, segmentation and genome annotation methods summarize a set of epigenomic assays, like the ChIP-seq assays. Existing methods output an annotation of the genome that assigns a single label to each genomic position. This strategy has some limitations. Instead, we propose a continuous annotation strategy that outputs a vector of chromatin state features to each position. There are some advantages of using continuous annotations. First of all, we can capture varying string of the elements. We can also capture multiple patterns of activities for each element. We preserve the underlying continuous nature of the input data. And finally, continuous features are very useful in visualizations and other downstream analysis. Thank you for listening to my presentation.